This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 229, recorded on April 19th, 2013. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiV, the podcast all about viruses. Joining me today right here in the TWiV studio on 42nd Street, Dickson Despommier.
Hello, Vincent.
I'd love to be in a glass-fronted studio and people looking in and watching you, Dickson, the famous Dickson.
No. They would be watching you,
But unfortunately, we're way uptown on the 13th floor, and all we can see is the gray sky.
We do have a glass window, though, and it's possible that the people in the apartment complexes across in New Jersey are using their telescopes to actually peep in on the TWiV shows. We don't know.
I'm sure.
We don't know that.
I always wanted ceiling-to-floor glass, but fortunately not the case. Also joining us today from Western Massachusetts, Alan Dove.
Good to be here, and especially good to be in Western Massachusetts today.
Here, so Boston has been really active today, right?
I'm on the unfashionable side of the state. If anybody's planning anything, there's nothing of interest here at all. Just keep moving. We're 90 miles from Boston.
You didn't. I'm glad you didn't have to go into Boston for any work this week, right?
Well, my wife had a conference she was supposed to go to, but she didn't feel right going after the events on Monday, so she canceled. And now, of course, it's Friday as we're recording this, and the whole city of Boston is locked down, and I'm sure her conference would have been canceled anyway.
Also joining us today from North Central Florida, Rich Condit.
Hi, fellas. How you doing?
Hey, Rich.
We're okay.
How's the weather down there?
I can say fellas today because Kathy's not there.
Have you had any severe weather events recently?
We had some pretty heavy-duty rain within the last week or so. But, no like tornadoes or anything, no. And today it is actually mostly cloudy and 75 degrees. Right. So it's good. The weather's been good.
Yep, okay.
17C here in New York. It's cloudy. Pretty humid today, isn't it?
Yeah, it is. It feels like late spring rather than early spring.
Rich, you have some family in Boston. They okay?
I do. They are locked down. They are okay. Everybody is okay, but they are, the place is closed. All the public transportation is shut up. We talked to our daughter this morning, and she and her husband and her two kids are at home, and they're just hunkered down till this thing blows over.
Wow.
Yep.
What a story.
It's an amazing story.
It'll resolve.
Yeah, they live in West Newton, which is pretty much adjacent to Watertown, where the center of the accident is right now.
Yeah.
Hope it all works out.
Pretty crazy.
Yeah.
You got it.
Well, we're here to talk about viruses. I know.
Good idea. Actually,
we're going to lighten it up with viruses.
Speaking of studios, when they have their fund drives, I pitch for NPR. So I was in a studio earlier this week.
Nice.
And actually, we got into a virology conversation.
Really?
And one of the things that the conversation morphed into is that science transcends politics and borders and that kind of stuff for the most part. So I think that in stressful situations like this, retreating to science to some extent is kind of refreshing. Okay, let's talk about something that is pretty much apolitical.
Yeah.
The thing is, the funding for science isn't apolitical.
That's true.
Or whether to work on life-threatening viruses.
As
whether
Tony
you think
Fauci said, right, once these guys in Washington sort out their political differences, we can get money to do what we need to do.
True. So, from that point of view, yes, there is politics, but the viruses don't know about that.
Right.
All right, our first story before we do some follow-up. Hillary Kaprowski has died. 96 years old.
Dear.
I would call Dr. Kaprowski a virologist.
Yes.
He, of course, was one of the people who made a attenuated polio vaccine in the 1950s.
How interesting.
And he, in fact, tested this vaccine in Africa and was blamed at one point
That's
in
right.
his career for having started the HIV epidemic.
Because
They said he grew it up in chimpanzee kidney cells, which had HIV in them, and then he fed it to people. But, of course, that's all wrong because now we know the virus clearly originated in the first 3 decades of the 1900s.
And even before we knew that, the evidence that was strung together was really kind of a tortured argument to
So he worked
pin it
on
him.
SSPE, didn't he?
I think he worked on a lot of things, rabies.
I knew
Yeah.
someone who actually worked with him. Dr. Michael Katz was one of his postdoctoral fellows,
Let's see, there are a couple of things I wanted to mention. He was here in 2005 to give a history of science lecture, which was really cool. And I have a photo, which I will post a link to, of the two of us, actually he, me, and Rich Kessin. You know Rich Kessin.
I
don't
gave
you?
a lecture in that series.
In 2005. And I remember after the lecture, he had been driven here by his driver. And he said, Vince, let's go have a drink. So we went to a bar. And we walked there. He could hardly walk. And he had a Boodles martini.
Nice.
And the bartender was like, what? I haven't had that called for in a long time.
But he had boodles. I'm sure he did.
He was very offended that he had been accused of starting the HIV.
I don't blame him.
I
He
don't
said,
either.
can you believe this? They're telling me I caused HIV. I can't believe it.
Right.
So I really enjoyed that.
This is a
Now,
nice picture.
isn't it cool?
Yeah.
It's good.
Now, he wrote Roger Vaughan wrote a biography of him called Listen to the Music, The Life of Hillary Kaprowski, and he signed it for me to my friend Vince with warmest regards, Hillary, 4/16/05. Wow, it's like...
Hold on to that. It's a collector's item.
Yeah, I'm going to scan this and put it on the blog.
Oh,
But,
great,
I
great.
just wanted to read one paragraph from the first. It was actually a pick many years ago.
Uh-huh.
Actually, the book is also signed by Roger Vaughan, who is the author. He says, for Elizabeth. I bought it off of Amazon as a used copy, and it had been given to someone else, and I guess they sold it.
Oh, I'll be darned.
Anyway, so this is very cool. So in the 50s, Kaprowski was experimenting with attenuating polio by passing it through mice. He passed it by intracerebral inoculation. And the book begins with this. A cold day in January 1948 at Letterly Labs in Pearl River. And he and his technician are bending over a wearing blender. Thomas Norton, a tall, stooped man, flips on the blender. He and Kaprowski frown at the machine as it whirs with a shrill scream, clunking now and then as the blades reduce chunks of solid matter to puree. They're grinding up rat spinal cord and brain tissue because they've been infected with the virus, and they're trying to make an attenuated virus. Kaprowski carefully pours 1 cubic centimeter of the cold, grayish suspension into each of 2 small graduated glass beakers. It is an oily glop. Brains are full of fat. The 2 men pick up the beakers and for a brief moment study the contents. It makes cold lamb gravy look appetizing. The material has been filtered for bacteria, but that is all. Heating it would have destroyed the virus. Norton and Kaprowski regard each other over the rims of the beakers. They drink, tipping their heads back for a long moment until the thick stuff flows sluggishly across their tongues. They make bad faces as they suck the greasy soup out of the corners of their mouths and swallow repeatedly.
I'd rather talk about Boston.
Kaprowski notes that the raw vaccine tastes like cod liver oil. When he can speak, Norton asks, "Have another?" Better not, Kaprowski says. I'm driving. I love it.
I want a Boodles martini.
I love it. That's just great, isn't
Holy
it?
cow.
Yeah, that is great.
My goodness.
And Matt Freeman sent me an email this morning. I met Hillary last year when he gave a final lecture at the Institute of Human Virology here in Maryland. He's old friends with Gallo, apparently. He told a hilarious story about why he didn't feel bad that the vaccine for polio is called the Sabin vaccine. He said he would hate for parents to take their kids to the doctor and ask for the Kaprowski vaccine and then make kids cry when they got it. He would much rather cry about Sabin, make them cry about Sabin than Kaprowski. Nice to meet him before he passed, a big name and good man. So his vaccine and Sabin's were put head-to-head in a monkey test mediated by Joe Melnick. And Sabin's won. They'd caused less disease in the monkeys. But it was really close. And who knows...
This last paragraph in the Washington Post obit is cool. Dr. Kaprowski was born in Poland, a talented musician. He was a penniless immigrant in Rio de Janeiro, making money teaching piano before joining a lab there and eventually moving to the United States.
Yeah.
Cool.
So I recommend this book, The Life of Hillary Kaprowski. It's really good. It goes through his whole life. It's got cool pictures in it. And
Excellent.
You will learn all about this guy who was pretty...
It was very influential.
Pretty influential, yeah.
He was.
He had unorthodox ways of doing things. When you went to his office at the Wistar, which he founded,
Yes.
he would have 2 martinis sitting on the desk. Old school
Oodles
stuff.
martinis, right?
One for
probably.
you, one for him. That's
Yeah, and so the book tells us about all this stuff. He was a little inappropriate. He would tell women they look good and stuff. So that's cool, but I'm sorry he's gone. But I'd wanted to do a podcast with him, but he was just too frail for many years. So, and 2005 was just before I had started doing it, or else I would have gotten. So,
Yeah, I also met him, I think in '05. I went to an event that was honoring Maurice Hilleman.
He went to that one, yeah.
Yeah, I was mainly there to see Maurice, but
yeah.
I've got a picture of the where I was sitting, the view of the table place markers. Just this all-star cast of virology right in front of me. It was amazing.
Yeah, it's very neat. All right, we have some follow-up. Liam writes, "Hi guys, I wanted to send you my thoughts on the HeLa sequence controversy. The entire HeLa sequence was and remains available from other studies. For example, much of the ENCODE project, the most thorough mapping of the genome to date, was done in HeLa cells, and that data is all available online. EMBL did their sequencing to establish a reference genome for HeLa cells. Generally, researchers aligned sequencing results to the reference human genomes, but as their paper documented, there are so many alterations to the HeLa genome that it is different enough to merit its own reference genome. I don't know how this should impact the discussion, but I do think it is important information. Whatever your feelings about the data takedown, this raises a lot of questions for proponents, myself included, of open access and open data publishing. I always like it when you guys discuss these pan-discipline topics. Keep up the good work. P.S. I kind of liked it when you called follow-ups FUs, and I'm surprised anyone took offense to the implication of a swear word in an informal conversation amongst adults."
Well, should have really referenced
adults in quotes there.
Should have referenced
Right.
Felix. Should have referenced Felix Unger, with an asterisk.
Yeah. He's putting adults in quotes because of you, Dickson. Because
Really?
you're beyond adult. You're like ancient.
This is a post-adulthood.
Yeah. Right. So I know the sequence is there, right? So, but still, EMBL should have probably contacted.
It's just as a courtesy, if nothing else.
And if the sequence is already there, then why sequence it? Obviously, they were contributing new information.
Yeah.
They were contributing a reference genome, and they billed it as such in their press releases and in the news coverage that they wanted for this. Hey, we sequenced the HeLa genome, and if you're going to do that, you need to ask.
True. I have a question about that, and that is how different from lab to lab, since you've been maintaining these cells for quite a long time now, how different are these HeLa cell?
That's a really good question.
Here's a good story.
Yeah, really different.
Here's a good story. So I brought HeLa cells to my lab over 25 years ago from the Baltimore lab.
Right.
And had been growing them all those years. They made nice flat monolayers.
Yep.
We grew them in spinner.
Yep.
We lost them last year. Okay, our liquid nitrogen died. At the same time, the spinner died because the spinning thing broke over the weekend.
Oh, jeez.
So I bought a new vial from ATCC. They're beautiful. They totally look different on monolayers.
Yeah.
Different.
They don't flatten as much. They make nice plaques, but they're totally different.
Are people keeping track of all these things?
No.
Because
We got HeLas that we've been growing in suspension for years. I've never actually tried to put them in monolayer. I should do that. Do you just stick them down in regular DME or something like that,
So we
Vincent?
spin them out of the SMEM, and we bring them up in DMEM and plate them, and the next day they're flat and beautiful. As Alan
I'm
will
going to try that.
Yeah. Yeah, and I remember, in fact, when I was in the lab, somebody, I think it might have been Michael, was trying to troubleshoot everything. 99% of experiments fail, and he was having some kind of trouble, and somebody suggested, well, maybe try a different line of HeLas.
Yeah.
And so he actually ordered a vial from ATCC, and they looked different.
Right.
And at that time, and that was 10, 15 years ago, It's it's like sourdough. Everybody's culturing these in continuous culture, and it's a cancer cell line.
Well, the next question then is, of course, how does the virus behave? Any virus behave in those cells then?
So there are two lines of HeLa you can buy from ATCC at least. One is supposedly better in monolayer, and rhinos tend to like that. There are lines of monolayer derived HeLas where rhino does better. Rich, tell me if they played out well and flatten out, because I'd like to get an aliquot. If they do,
I'm very curious.
I'd like to get an aliquot because I don't like these that they don't flatten out.
Really,
it's hard to detect viral CPE, which we used to detect by seeing the flattened cells round up.
Right.
But they're all round to begin with.
So I've never tried that with these. I got them from Flanagan some time ago. But, they've been, spinning for a long time. Well, actually, we just thawed some out recently. I don't know. So they got a complex history, but I'll try this.
These are handed down from lab to lab through different lineages, and they're all going to be radically different.
We've had this discussion before about other things, but here, I'm an outsider in terms of virology, but I'm not an outsider in terms of strain differences and animal differences, et cetera, et cetera. So you've got the same virus in the same cells. No, not really. You've got different cells, and it's almost like the same virus, but it's not quite. And you're both doing the same experiment. You both get different results, and you have arguments at meetings.
Yeah.
Oh, very often it's a cell-based thing, yeah.
Right.
You have to be very careful.
Of course you do. So how do you be careful? The point is, how do you be careful?
Well, you cannot base everything on what you find in HeLa cells, for one.
Do you trade cells and say,
absolutely. You trade cells. When you can't reproduce something that some other lab published, you get through reagents.
For example, we study innate immunity to polio.
Right, sure.
We're doing it in HeLa cells.
Exactly.
If we're really serious, we're going to do it in a respiratory epithelial cell line, and that's what we're
primary explant. You mean a primary explant?
Well, primary, you can buy them, primary explants. You can take stem cells and differentiate them into lung respiratory cells. Yeah. So we're going to test what we found in HeLas in these relevant lines. There
are actually several companies now offer primary lines.
Sure.
And they even specify the population that it came from.
Right.
But so these are not immortalized cells.
No. Primary cells.
Right.
So it's a limited supply, but they'll give you some information. They obviously it has to be kept anonymous, but they'll give you some information. This is from a Caucasian male. This is from an African American female, because those are going to be different too.
Absolutely.
Yeah, they do procedures medically where they get these cells and they sell them.
I got it.
There are not a lot of them, but you can get them. You can buy them. They're not cheap either. Expensive. All right. Catherine writes, Annyeonghaseyo Twivers. That's hello in Korean. I'm very sorry if I butchered it. Okay. All is well this evening. The cherry blossoms are out, and we're expecting highs in the upper 60s, about 19 Celsius. On TWiV 227, Vincent and Kathy discussed a figure showing the sialic acid binding sites in the human respiratory tract, including the eyes. With the outbreak of human cases of the bird virus H7N9, is it possible viruses getting into the respiratory tract through the eyes? As they described in the figure, there are alpha-2,3 receptors in the eyes. I can envision a situation where a person is killing a chicken for a customer and blood or other bodily fluids spray into the eyes or inhale. Just some naive thoughts. Actually, not naive at all.
No, not at all.
I emailed Ron Fouchier.
Well, they were naive because she didn't have this background, but not a silly question at all.
I emailed Ron Fouchier, and within the hour he had responded. He gave me a long email, mostly with unpublished data, which I can't share. But he said, yes, there are alpha-2,3 sialic acids in the eyes, and yes, it has been postulated that the eye may function as a port d'entrée for some flu viruses. Belser and Tumpy have done great work to follow up on that.
Wow.
And I'm going to put a link to two papers in the show notes. One is called Ocular Tropism of Influenza A Viruses, Identification of H7 Host Responses. So basically, these H7 viruses love to replicate in the eye. Mm-hmm. And there have been, in outbreaks of avian H7 viruses, a lot of the poultry workers get conjunctivitis as the virus is replicating in the eye.
Wow.
So then it could spread to your tract. So that nasolacrimal duct, that goes right to your nose. So
yeah. There's another worry there, too.
What's that?
Well, that is the optic nerve. That goes right back into your brain. So if the virus is replicating...
Not an especially neurotropic virus.
Yeah, right.
Actually, enterovirus 70, which causes conjunctivitis, in 1 in 1,000 or so cases, goes back via the optic nerve to the brain.
Thank you, Vincent.
You got it, Dickson. You're absolutely right.
There's in one of the articles that we're coming up to much later, there's an interesting little line that says that taking eye swabs is a better source of diagnostic material than nose and throat swabs.
Interesting.
Right. That is a Fouchier article, and that's because in outbreaks, these people make a lot of virus, apparently for H7s, not the other.
For H7, right.
So,
it replicates a lot in the eye. However, Dickson,
yes.
they're not seeing conjunctivitis in China.
So I have a question about that too. Then, I mean, what are the antiviral properties of lacrimal secretions?
Oh, I have no idea.
But there
There
should
you go.
be.
Write a grant.
Has anybody looked at this?
I'm sure. There's a huge literature. In fact, Tumpy et al. Have done lots and lots of work with flu. I'm sure there's an innate response there.
Because there
There
are lots
are
of
intrinsic defenses. Well, tears themselves, of course, are an intrinsic defense.
There are Meganins in tears as well.
Is that right?
Yeah, sure.
So I'm sure there's a whole field. I have never explored it,
All right.
but we could.
It seems to have risen to the
A
top of
quick
our list.
PubMed search on antiviral lacrimal secretion only returns 6 hits, but
That's
that may not be
all?
the.
Well, that is cause for writing a grant.
You want to get back in science, Dickson?
I'm back in it already. We'll discuss that later.
All right. The next one is from Stephen, who sent us a link to a New York Times article on the H7N9 story, and he says, "I just wanted to offer a word of praise for Denise Grady of the New York Times for including these sentences. Quote, researchers are not sure how deadly it is because they do not know how many people have been infected. The worst cases become
Bingo.
obvious, but if some people have mild symptoms or none at all, many cases could go undetected."
There you go.
Yes.
Good for her.
Now,
Yay!
I have to say that during the H5N1 brouhaha, Saul Silverstein, my colleague here, is a friend of Denise Grady. And he read one of her articles where she said, the mortality rate is very high. And he said, look, this is why you're wrong. And now she listened to him.
How about
So
that?
that's why she's
How about
doing this, because of Saul. So thank you, Saul. Just making a funny accent there. Researchers, he continues, he sends us a link to an NPR story. He says, Nancy Cox, a CDC flu person, notes the virus already has 2 genetic changes that enable it to infect human cells and reproduce at human body temperature, which is lower than birds. Right. We'll talk about those in a moment. That means, she says, these viruses may already be partially adapted. Not totally adapted, but partially adapted towards the type of virus that might efficiently transmit from human to human. This comment by CDC's Nancy Cox, as quoted by Richard Knox on NPR, brings up a question. This is Stephen writing now. Let's say 5 changes are necessary for a wild-type bird flu virus to become a dangerous human flu virus. Reporters usually write as if genetic change 1 predisposes the virus to acquire change 2, and 1 and 2 make it more likely that 3, 4, and 5 will occur. They also usually report that such changes are always one way. Once 1 is acquired, it's never lost. Is this true? Is it any more likely that a virus mutation 1 would gain mutation 2 than it would lose 1? Is Cox's implication true? Do the mutations for a zoonotic infection make it more likely that the new H7N9 would become able to transmit between humans?
Very good question.
Well, my view is that these mutations are accumulating randomly in birds somewhere, right? And if they contact a human and it's the right combination, the virus will replicate in people. There have been lots of contact between H7s and humans over the years, and not all of them result in outbreaks. What we have now is a situation where we have 2 mutations. There's a HA mutation that allows adaptation to human receptors, and there's a internal protein change that allows replication at the higher temperatures of the human respiratory tract. So these probably are important for the virus to replicate in people, and it's why you're getting transmission from birds to people. But it's not transmitting from person to person, and that could be a mutation selected for in a person. It could arise randomly in a bird. So if it arises in birds, there's probably no sequence of events. It's just random. And then it gets into people and it transmits. So I don't think there's any necessary sequence, but you do have to have changes that allow it to get into people in the first place.
The changes you'll see and the order they occur will depend entirely on what maximizes the virus's ability to spread to new hosts.
Right.
That's exactly
Right, and you have to kind of let go of the idea of predisposition.
Right.
I mean, there are things that can kind of look like that because, there is a sort of a karma associated with this, where if you get a foothold under one circumstance, then it sort of changes the selection pressure relative to what it was before that.
Right.
Okay, and that may help select for certain events that happen, and it's sort of that may kind of look like predisposition, but it's not.
Yeah.
Well, for example, if the virus jumps from a bird into an individual human, and it had a couple of mutations that allowed it to do that, that virus population in that patient is now under strong evolutionary pressure to find a new host.
Right.
And that may, if it acquires mutations that allow it to replicate better in humans, that would increase its odds of finding a new host. But we don't know what the constraints are on that and whether it's really capable of developing those mutations in the course of a single infection cycle.
Yeah, I think this is important, that if things can arise in people, say, that would allow it to transmit, but they have to be compatible with the fitness of the virus.
Yes.
Because they may not be. And I think here, virus has been infecting people since February. It's been mutating and mutating every replication cycle. I think it can't transmit. I don't think it's compatible with this virus's replication cycle. Now,
also, from the virus's perspective, it doesn't have to find a new human host. It just has to find a new host.
Yeah, that's right.
Right.
So if it's a human who's a poultry worker and they picked it up from a bird and it can now reinfect another bird, yeah, that's fine.
I don't think it's trying to find. It's just replicating, and it gets expelled in the air. And if it's adapted to transmission, it will. If not, that's the end. But the virus is elsewhere.
It's a random deal.
It's random. That's the key.
You'll select for mutations that give the virus more ability to replicate in some host.
Right. Dickson, did you?
I had it was a related question. I was just wondering how tightly bound the receptor molecule is to the HA. How is it? In other words, if you were to flood an area with a free sialic acid, would this prevent the virus?
I asked Peter
penetrating?
Palese that. He says it's not good enough to prevent infection.
That's
why we haven't developed sialic acid analogs that do that.
Exactly. So it's constantly seeking out, and then
it's bound and then endocytosed, very quickly.
Quickly.
Yeah.
But in vitro, if you were to just flood a solution of virus with a particular version of sialic acid,
you can compete in cell culture for sure.
But
Peter said that they're not therapeutically useful. It's been tried.
Okay. But once it binds to the virus, does it come off again? I mean, is there a kinetics
the
to this?
neuraminidase will. I'm sorry. If you use the soluble sialic acid analog, would it come off again?
Yes. That's what I'm asking, actually.
Apparently, it just comes off enough to make it not therapeutically useful.
So there is a kinetics to this.
Yes, of course, as always.
It's not a permanent binding.
Ligand receptor interactions are always dynamic. They never, very rarely stay on forever, right?
Correct.
Well, and in fact, saturating receptors or saturating viruses to prevent receptor binding, people have tried that with a bunch of different viruses, and I don't think it's ever been very successful. You either have a kinetic problem that prevents it from working in the first place, or you get it to work, and then you get escape mutants.
Yeah, I think these blocking receptors is, in general, not a great therapeutic. I think part of the problem is you have to block every virus.
I know.
Yeah.
So I'm trying to make bridges between your science and my science. So
What science? Your science is
parasitology.
Vertical farming?
No, stop it. No. Entamoeba histolytica in culture will kill a Chinese hamster ovary cells. All right? And it binds to them by galactose. There's a binding. It's just like this. It's the same.
Okay.
But if you flood the culture with galactose first and then put the amoeba in there, they do not. They live side by side, but next to the cells. They don't kill them at all.
Okay. No,
So you can prevent in killing
culture,
or
you
invasion.
can do the same thing with flu.
It's
but,
the same. Okay, fine.
There are
you have
plenty
to.
of experiments, but
even,
therapeutically, it is in people. It's not good.
No.
Right. You can prevent it from infecting a cell, but you have to practically put it in a crystal of the receptor. You have to put so much concentration in; it's not practical.
Oh,
yeah, because then in your respiratory tract it gets diluted. And,
no, but of course,
right.
but of course.
One more question from Rui. It says, he wrote this to my blog, but I thought it was a really good question, so we'll read it here. I'm a student from the University of Edinburgh in Scotland, also a regular reader of your great blog. I actually have some quite stupid questions to ask you.
No, you don't.
They can't be stupider than the ones I just asked.
No questions are stupid here on TWiV.
Okay.
These are particularly not stupid.
Oh,
I read
very
some
good.
news, for example, this one in Nature. Basically, people are concerned about human-to-human transmission of the novel H7N9 avian influenza virus. But what I am confused at is the definition of human-to-human transmission of a flu virus. What is required for a flu virus to be transmitted effectively in humans? And what is the boundary there lying between the zoonotic transmission and human-to-human
Oh,
transmission?
those are fabulous questions.
Does human-to-human transmission mean wherever the flu virus acquires its segments? 1. Certain types of cells are permissive to flu virus. The virus can enter the cells and replicate efficiently. 2. The progeny of the virus is able to be present in the aerosol produced by the human host, which is able to infect the other individual. If I have a strain of H7N9 virus, I infect human A549 cells, which are lung cells, and get a very good titer from plaque assay. What does this mean? Does this only mean A549 cells are permissive for this H7N9 virus? This is an interesting question because there's a fellow on the floor here who's working with avian influenza viruses, and he says they infect human cells just fine. But they're cells in culture. They're not people.
Right.
And they're not being transmitted.
Right.
What you need to transmit among humans is anybody's guess.
Right.
You know, we know a little bit about ferrets for H5N1, but my guess is you need to make a lot of virus in the tract. It's got to get into droplets. It's got to be stable. It probably has to have a certain fusion optimum, right? It has to induce coughing. All these things probably matter. So
That was actually the crux of the question that the Kawoka and Fouchier papers we were talking about a few months ago were looking at, is what do you need in order to get transmission from mammal to mammal of these avian flu viruses?
And the results were very interesting because they found that the pH of fusion really influenced transmission, which is not something anyone would have guessed. So I think an interesting corollary to this is how would we know when it's being transmitted from human to human?
How would we know, Vincent?
Dixon, can you, for one?
I can certainly predict
Go
that
ahead.
you're going
How
to
would you
get
know?
an explosion of infection in one particular place, which then spreads out to the countryside.
Epidemiologically linked, right?
Absolutely.
Families all getting infected, clusters, right?
Yeah.
And what
Health workers.
What about if you could sequence all of them? What would it tell you?
Oh my goodness! It might show the difference. It might say why it's happening.
So what you do is you sequence the isolates and you construct phylogenetic trees, and you
exactly.
can make a lineage. You can say this virus came from this, from this.
That's right.
Whereas right now, and in fact, one of the papers that we're linking to has a very nice phylogenetic tree of the existing viruses in China, the H7N9s. The human viruses are all at the ends of the trees, which means they're all at last point infections, but they don't give rise to any other infections. You could tell that by phylogenetic analysis. You okay with that?
Actually, there have
Very
been
much.
several pandemics that are reasonably well characterized. Has this sort of analysis? How good is the similar analysis for those pandemics?
Right.
I know that people have had an understanding of where the segments came from for a given pandemic, but how detailed is that? Because there is a history here where you might be able to get some idea.
Oh, there's tons of 2009, especially the last pandemic, and we had lots of sequence
In Mexico.
capability.
Yes. Mhm.
You can see these strains diversifying in populations, and you can see clear transmission patterns for sure.
Mm-hmm.
It's beautiful.
Yeah,
Right.
2009 was incredibly informative because it was the first pandemic where we had all the tools to really do these detailed analyses of it.
Yeah, and you can say these two people in this family got infected with a virus, and here's the ancestor, the predicted ancestor, very clearly, and on, and the viruses are in the middle of trees rather than being at the end of the branch.
So, Vince, that's viral history. The biggest CDC question, and everybody else's, is when is the next one and where is it going to occur?
Well, yes.
And that's what this can't answer.
That's why they're paying attention to this. They
But of
want to know
course.
if this is the next big
No,
one.
no, I got it.
I don't think it is because it's been circulating and it's not transmitting.
Right.
Let me ask you guys a question, okay? Do you know any example where a virus went into people, replicated for a while, and then acquired transmissibility by getting a mutation? Can you tell me an example of that?
I know of none. Okay, that doesn't mean one doesn't exist, but I personally don't know of any.
I can't think of any. You know?
Well, yeah, not that we know of, but there's
Not that we know of, right.
the early history of many viruses is pretty murky.
Well, yeah, so 1918, there's this hint that there were waves of increasing pathogenesis, but we don't have any sequences to prove that.
Right.
And yes, it may have happened, but...
And when polio entered humans, what went on? When hepatitis C entered humans, what went on? We all the viruses that are in common circulation, we just we don't have good data on.
I mean, HIV even, we only have recent isolates, which are pathogenic.
There are many.
Yeah, so I think we don't have that information at all.
You getting impatient, Dickson?
Not really. I just want to say something, because there are many zoonotic viruses that were never in humans before that suddenly got into humans that could easily be transmitted from human to human. And I'm thinking now of Lassa fever, for instance, and some others like Ebola. Those
they don't transmit.
How do you think the other people
They don't transmit from human to human. Ebola does not transmit from human to human.
I thought that's how the health workers got sick when they were taking care
Yeah,
of
when
them.
you bend over someone and they cough in your face. And,
That's transmission to another human.
No, it's only close contact mediated. It doesn't go aerosol.
Oh, you have to...
That's why there are no huge outbreaks of Ebola, because it's restricted to the hospital and the houses where the families are ill.
Oh, I was using a totally different explanation for why there are no big outbreaks, because it's so severe and so fast that it's quarantined immediately and nobody else comes into the area.
That may be. But Reston is not pathogenic and it hasn't spread in people.
No,
either.
but I all right.
Yeah, if somebody with an Ebola infection takes the bus and you're on it, that's not going to give it to you.
No,
but
you have to.
It could have happened with
Lassa.
Lassa. It could have happened.
I mean, unless they're standing right in front of you and they cough on you and there's some blood and you have really direct contact. But
now you're stretching the definition of human to human contact. So I think you should define
aerosol transmission. That's
period.
what we're saying. Okay,
no, that's fine.
Ebola does not
that's
transmit.
Fine. No,
by
that's
aerosol.
absolutely fine.
And these infections in China
I'm okay with
are not transmitting human to human by aerosol, or
That's
in any
fine.
way. There's no
No, that's
epidemiological evidence that there's.
for flu, aerosol transmission would be the standard. For other viruses, you need a transmission mechanism that will actually sustain an epidemic. Is the issue.
Yeah.
So all I'm saying is, this virus could acquire other changes that allow it to transmit through the air, and it would be the first time we've actually seen this happen.
Got it.
So far, with all our tools, we have not actually documented the changes that are needed to, say, acquire aerosol transmissibility in humans. Mm-hmm. And I am suspicious, because I think this virus has been around for a while in poultry. It's been mutating like crazy, and it doesn't transmit. I don't think it's compatible. In fact, there have been some other outbreaks of H7 viruses, and they don't transmit well. So,
which we'll talk about in a moment.
So let's go over the current situation. Now the papers are starting to come out.
Yeah.
Right? New England Journal, Eurosurveillance, all over the place. So far, as of yesterday, 87 cases, 17 deaths, 5 people got better. That's 20% case fatality rate.
Right.
Last time we talked about it, what were there, 6 cases? This is 2 weeks ago, right?
Yep.
Yeah.
So
what did you say? 87 cases and how many deaths?
17 fatalities.
17 fatalities.
So case fatality rate's 19.5%.
So we've got 70 people who have recovered.
Yeah.
What are their geographies? What about the epidemiological stuff here?
I guess.
They're scattered around China.
Age groups?
Oh, hold on, Dickson. Just a minute. Let's look at where they're all over China. There actually is a great map at WHO.
Yes. Right.
Let's bring that up. So WHO is putting out weekly summaries.
Good.
So here's a map of China:
I can see it from
Anhui, Zhejiang, Shanghai, Jiangsu, Beijing, and
Is this the one from April 10th?
This is the latest one. Is April 16th? Is the link I put in the show notes is April 16th. So
That's pretty
that's
up to
this
date.
week's. So you see, Dickson, it's
Yes,
quite.
I see it.
This is a big area,
Yeah,
even though it's not in the west yet, right? It's all on the coast there, and a little
So
bit north.
that's another reason for believing that it's from animal to human, not from human to human.
Why is that?
Yes.
Dickson?
Well, because it's so random in terms of human cases.
But that would imply that the birds are
There's no
infected
epicenter.
all over the place.
Yes, it
right?
would. Yes, it would.
Yeah.
And now the thing to point out is this is a low pathogenicity virus, so that you don't often know that the birds are ill.
Right.
Right? They get depressed, but not.
Just a geographical footnote. You mentioned there aren't any cases out in the West. That's partly because there isn't much of anything out in the western part of China.
Right. No, there's no birds out there.
I mean,
That's right.
there are massive, completely unpopulated areas, and then there are massive areas that have scattered nomadic tribes in them. So the virtually the entire population of China is concentrated in that eastern portion. Some people have described China as an island that just doesn't look like an island.
It's a big city.
Because the western part is completely walled off from the rest of the world by these vast expanses of empty landscape. Not entirely empty. There are populations in it, different ethnic groups that live out there.
They're flocking to cities like crazy because of
they're very poorly covered by epidemiology too.
But of course, out west, at the border of India, Pakistan, there was an outbreak of polio not too long ago. So
yes,
remember that?
Right.
Yeah. So there are people out there, but I don't know if
not too many.
I don't think we'll be seeing reports of this for a long time.
So this is an interesting point, Dickson. If you have this widespread cases and there's no human-to-human transmission, the implication is that birds are giving it to people. But of course, it must have spread throughout. I mean, the presumption here is it's poultry, right? That's sold to people.
Exactly.
And somehow the birds are infected.
Exactly.
They are doing a lot of swabbing of birds over in China. Let's see, they have done how many thousands of swabs? Over 48,000 test samples from live animal markets, farms, and slaughterhouses, and only 39 so far have been positive.
Really?
And
now, the other question that comes up with this distribution is if it is jumping, if these are just individual jumps into people, why now?
Good question.
Well, and I'm looking at this on that same link with the map from the World Health Organization, Vincent. On the bottom, they have the epidemiological curve of confirmed cases.
Yep.
And there's a peak that then tapers off. Is that because the data is there a lag in reporting or something that causes an apparent drop-off, or is it actually declining?
It's a good point. I wondered the same thing because, yes, there is a peak around April
2nd, 3rd, first.
And now we're at this. So this data were as of the 16th. So there have been a few more cases, but it looks like there's a peak. Which may be now there may be incubation periods that are delaying. I don't know.
Or they could have killed off all the chickens.
Yeah, exactly. There's been a massive cull of poultry in China at this point. So
the meat markets are shut down.
There may just not be sources for it at this point.
But where it actually is kind of puzzling. So they're doing a lot of sampling, and there's an article here at ProMed Mail by Helen Branswell, where she talks to our buddy Mike Osterholm, who says he wants to see exactly what they're doing to test for the viruses in China because he's not sure they're doing it right. That's basically what he's saying, right?
Yeah.
So that's interesting. Let's see. Bunch of cool articles here. So now the sequences of 7 different ones are being deposited, have been deposited. There was a New England Journal paper that analyzed them, and also an article in Eurosurveillance.
And what did they find?
Okay, let's talk about that. So the sequences are from the first 3 cases. They're called A Shanghai 1, 2, and 3, 2013. Then they got a sequence from Hangzhou, from a male patient. Then they got sequences from a pigeon, a chicken, and an environmental sample in Shanghai. Right. These are from the Shanghai market. Okay.
And they is first author is Gao. The last author is Xu, and there must be 20 or 30 people here, and it's all from the National Institutes for Viral Disease Control and Prevention, Chinese Center for Disease Control and Prevention, Key Laboratory for Medical Virology, National Health and Family Planning Commission. They're all in Beijing.
Right.
Yeah, it's actually quite a nice article. They really
it's a great article.
They must have been cranking out the writing, you know.
So they have the people.
And the important thing is, what I like is that this goes from the patient cases and the symptoms and all of the sort of clinical stuff through the sequencing and phylogenetic analysis. So it's a very thorough analysis of the whole thing. It's really a fun read, actually.
Then I think 2 days later,
and it's open access.
It's open access,
right? So you can have a look at this.
Another sequence paper came out in Eurosurveillance, which seems to be a collaboration between Japan and Kawaoka Lab in Japan and Wisconsin. So I'm going to sort of combine these two analyses, but they both come to basically the same or similar conclusions. All right, so first of all, Shanghai and Anhui isolates are very different.
Right.
So it looks like there's at least two introductions, but probably more. They're all triple reassortants.
Gee.
The HA is most similar. I shouldn't say come from, because you never know. When you sample today, you can't know the ancestor. You have to imply it on a phylogenetic tree, right? Mm-hmm. So the hemagglutinin gene is most similar to one from a duck, an H7N9 virus that came from a duck in Zhejiang. The neuraminidase gene is most similar to the neuraminidase gene from a wild bird, an H7N9 virus from a wild bird in Korea. And then the remaining 6 internal segments, the ones that encode for internal proteins, are most related to genes from a brambling, which is, I guess, a type of bird.
It's a small bird.
Brambling.
From Beijing.
I had to look that one up.
It's an H9N2. So this is a triple reassortant. Where that happened, who knows?
Wow.
But I think, it's random stuff going on. And, you get the right combination to infect a person, but then you have to be in the right place at the right time and infect the person.
That's right.
And this is just what happened.
Their suggestion is that the reassortment happened in an avian host, but they don't really know that.
Right.
No idea.
Could have been pigs. I mean, that's the earlier hypothesis, right? That's the mixing vessel that everybody was talking about.
So Shanghai 2, Anhui, and Hangzhou isolates, where they are over 99% identical, yet those cities are pretty far apart.
Yeah,
they are.
So I don't know. I guess poultry being transported long distances, right?
Poultry in motion.
It's true. Oh.
Shanghai 1 and 2 have 53 nucleotide differences. Those were both isolated.
Wow.
And so that's one of the reasons why it may be 2 different jumps, right, from people.
Yeah.
Now, there are a bunch of, when you compare the human isolates to the animal precursors, the presumed animal precursors, you find some interesting amino acid changes. And let's go through some of those. So basic amino acids at the HA cleavage site. When you have a multi-basic sequence at the HA cleavage site. Now, cleavage of HA by cellular proteases is required for the virus to be infectious. And most seasonal strains don't have a multi-basic cleavage site, and that makes them cleaved only by proteases that are in the respiratory tract. The highly pathogenic viruses like H5s have multi-basic cleavage sites, which lets them be cleaved by ubiquitous proteases, so they tend to be more pathogenic as a consequence. So these viruses don't have a multi-basic cleavage sites, and that's one reason why they're low pathogenicity.
As defined in birds.
In birds, exactly. All 7 viruses have a change in the hemagglutinin, which allows them to better bind to alpha-2,6 sialic acids. Those are the human.
Right.
Although we know that there are alpha-2,3 up in the eye and the nasolacrimal duct.
Right.
So I don't know how important that is. It probably helps. So that's one of the changes that Nancy Cox says is one of the things that makes this on the way to a human virus.
So, Vince, instead of tracing the virus, you could trace the chickens.
That might be hard, right?
I think it'll be easier to trace the virus.
Well, it depends on how the chickens are being raised, because I don't know much about chicken poultry sciences in China, but there may be some huge producers that then distribute through, this is my territory, this is your territory, and it looks like 2 separate strains came from maybe similar places.
Well,
my understanding is that farming in general in China is very differently run from the way it is here.
No, you're right. You're right.
It's likely to be a lot of small family farms, more or less family farms out in the countryside, and packing the chickens onto a bicycle and taking them into the city.
So there's a little note in ProMed Mail. The Hebei province has put 1.7 million chickens and ducks on or around Baiyangdian Lake under close observation. So they're looking at all these birds, all right? And then it's a major migratory route, which is why they're doing this. And then there's a note by the moderator. If previous reports from East China are to be relied upon, no illness or mortality will be seen in the migratory birds. To trace infection with H7N9, either the virus or antibodies should be discovered. So there you go, Dickson. They were thinking about your question.
Yeah.
What else? Let's see. Some more mutations. Okay, so sialic acid receptor binding. There's also another change in the HA that blocks glycosylation, which could improve alpha-2,6 sialic acid binding as well. They all have a 5 amino acid deletion in the stalk of the neuraminidase. And this is believed to be selected for during replication in terrestrial birds. So I don't know if that would affect people at all. One of the viruses, the Shanghai first one, has an amino acid change in the neuraminidase, which is associated with resistance to Tamiflu.
Right. When you said terrestrial birds, did you mean chickens but not ducks?
I don't know. Is it duck? What about gallinaceous? How about that?
Well, ducks are not terrestrial birds. They're water birds. Waterfowl.
What does gallinaceous mean?
That's just birds.
No,
That's a general
domestic
term.
fowl. Terrestrial bird, not ducks.
Not,
Like chickens and
not
turkeys.
ducks.
Okay.
Yeah,
Not
chicken,
ducks.
turkey,
Not
grouse.
geese, not ducks.
No, not those
Right.
guys are water birds, right?
I know that, but they're kept... Duh.
I don't know birds, Dickson.
No,
I'll take your
that's-
word.
Sure you do.
All it said, it was two papers. They said
Right.
terrestrial birds and gallinaceous birds, which is chickens and poultry, right?
Not passerine. The passerine birds are the ones that cling to trees and stuff.
All right, and now you're getting way above my pay grade here.
Yeah. We need to call the folks at TWiV.
This Week in Birds.
That's right.
What a great podcast. Make it. I'd listen.
Yeah.
So Tamiflu resistance, that is not good.
No.
Now, that must be a random thing that just popped up in the bird, right?
So what aspect does Tamiflu
I would assume
interfere
so.
with?
It hits the neuraminidase.
Neuraminidase.
It's the neuraminidase. Oh, brother.
Let's see. Oh, there's another change that's really cool. So PB2 gene, one of the polymerase subunits, it's glutamic acid to lysine at position 627. This is selected for when avian viruses go into mammals which have a higher temperature. This allows them to replicate better at higher temperatures, so it's a virulence determinant. So that's one of the others that Nancy Cox pointed out.
Huh.
And then there are a bunch of others. All 7 are resistant to amantadine, which most viruses circulating now are anyway. And those are pretty much all. There are more, but those are the big ones. But they don't transmit. What I thought was interesting was a paper which came out yesterday after I had sent Ron Fouchier this question about infecting the eye. Like 2 hours later, I noticed he had just published a paper in Eurosurveillance. They reviewed an epidemic of H7N9, not the same virus as this one, in poultry farms in the Netherlands 10 years ago.
Oh, yes.
And this involved 250 some odd poultry farms, and 453 humans got infected. 89 were confirmed to actually be infected. And during that outbreak, there was a lot of conjunctivitis in the people. There was one fatal case in that outbreak, and the virus from that person had the E627K mutation in PB2. None of the other isolates had that. So the suggestion is it may have evolved in that person. Unfortunately, it didn't go from person to person. So there was no human-to-human transmission in that. So no evidence.
Am I correct also that person had a predisposing medical condition?
Could be.
That is the case with a lot of these. All 3 of the people in the original paper from China had issues.
Right.
Not necessarily respiratory issues. 2 of them had hepatitis B, and another had, I forget exactly.
I noticed those were people who were really sick, so that could have been part of the reason. But I don't think everyone has had other medical conditions that have been infected.
One had chronic obstructive pulmonary disease and hypertension. 2 others had hepatitis B, and one of those was obese, which we have heard before can impact on things.
I bet this is sort of a pet peeve of mine now. I bet if you sequence the genome of all these people who got very ill, you would find polymorphisms in their
Factors, yeah.
in their immune genes that are predisposing. I think the early cases in an outbreak like this are typically kind of sentinels, which really are people who are immunocompromised in some way, but they do point out that there's something going on. It's like a canary in a coal mine, right, Dickson?
Yeah, it sounds that way.
Yeah, and I think within a few years we will be at the point where it'll be feasible to do those types of analyses on early cases in a pandemic. Because it's already, the cost of sequencing a genome is down in the few thousands of dollars.
Yeah.
This paper from China, the first case was in February, mid-February, and the paper was published on the 11th of April. The third case was in mid to late March. So this took them less than 4 to 6 weeks to do all this. It's amazing.
There was a comment on one of my posts by GSGS, who we've mentioned before. He says he's talking about H7. I think it was referring to the Dutch H7N9 outbreak that I just talked about. We said as it reached Germany, it died in 2003. Was it the summer weather that killed it? I think H7N9 won't go into wild birds because it's very poultry-like. You usually don't see such viruses in mallards or shorebirds. And he says the biggest danger, in my opinion, is that H7N9 would reassort with H1N1 or some human virus. So I don't know where that would occur, because I don't know if H1N1 is in the animals where H7N9 is. Well, I guess the surveillance should tell you, right?
Yeah, I'd be more worried about it reassorting with a swine strain, because that's probably an easier route to get into mammals.
So far, they have not found this in swine.
They haven't found it in pigs.
Yeah.
Even the ones that were washing down the rivers, which actually, there's something bad going on with Chinese agriculture right now. I'm not sure exactly what it is.
Well, there's plenty. Name it.
There's plenty, but this year featured the rivers running with pigs, and then the H7N9 jumping into humans. And I don't know if those are connected in any way, or if there's some larger weather event that has something to do with it. But it's an odd coincidence.
So I want to see if we can kind of summarize this at this point.
Good idea.
And I don't know if I can do this. Let me give it a try,
see if you've been listening,
correct
dude.
me.
Or reading.
So the impression that I get is that there are H7 containing viruses that are probably pretty widespread in birds and not necessarily very pathogenic, so we're not very aware of it. Mm-hmm. And there have been, at least over the last decade or so, and probably before that, but we couldn't pay the same kind of attention, there have been sporadic emergences of little pockets of infection of various reassortants of H7 containing viruses in humans that then apparently kind of burn out and go away. And the most recent thing we're looking at is another example of the same thing. In this particular case, a triple reassortment of an H7 and H9 virus and some other virus to contribute the internal segments. Is that about the picture?
Sounds right. These are low pathogenicity. In fact, the outbreak in the Netherlands was picked up because it was a high pathogenicity virus, H7N9. That particular isolate was highly pathogenic, and it killed the birds.
Highly pathogenic in the birds. And that's interesting because it was not highly pathogenic in the people.
No.
Except for that one case, they all got mild respiratory disease.
So you've got other things here too, Rich, which I would like to add to, and that is that you're right in the middle of the migratory season for wild birds, and there might be a seasonality to particular cuisines which require lots of poultry, for instance. And so you might have a surge. You might have some cultural aspects and some ecological aspects which superimpose on top of this. It sounds like that two unfortunate events, at least, and maybe more, occurred throughout China, which resulted in these emergences of these pathogenic strains.
Well, there is actually a rather ironic seasonal demand for a particular poultry product, the flu vaccine that's grown in eggs. That requires such a large quantity of eggs in countries where the plants are making this, they have to schedule with the farmers in advance to make sure that they'll have enough of a supply of fresh eggs to meet that demand. There's also, in Western countries, there's an Easter spike in egg use, but there are some seasonal demands on poultry products that might factor into the population of them at a particular time.
Ironic, isn't it?
Yes. Wouldn't it be ironic?
It would.
Yes.
Happy Easter.
If the requirement for more chickens to make vaccine actually
Helps foment the pandemic.
Here.
I think in the U.S., you make 150 million doses. That's 150 million eggs, roughly. I think that's a fraction of the egg consumption. At least that's.
It's not like tripling production of eggs. I don't think, but it is a significant enough difference that they have to make sure they get their orders in, and
I think
poultry farmers have to adjust.
We do need to move away from using eggs to make.
Oh,
vaccines.
absolutely, for many reasons.
We could raise them in plants.
We're moving towards it.
Yep.
And vertical farms, vaccine farm.
There are 3 now.
Cell-based vaccines are now being approved. So.
There are 3 factories just like that now in existence.
To do what? Make vaccines?
In plants.
Yeah, I think it'll happen.
Yep.
Things go slowly.
Yep.
So that is the situation. And last time we thought this was not going to be a pandemic strain. I still do not think it will be.
It doesn't sound like it.
Doesn't sound like to you. You guys, what do you think?
I do not believe that this will become a pandemic strain. I'm impressed, however, with I have to pay attention to people like Nancy Cox and others who are responsible for, on a large scale for monitoring this kind of thing and making sure that people are safe and they are approaching this with appropriate caution.
Yes.
So you
making
think
vaccines and that kind of stuff.
You think they're doing the right thing?
They don't seem to be freaking out, all right, which is the right thing. But they've identified one of the. I think going ahead and making a vaccine is not an inappropriate thing to do. Right. So they're exercising caution without freaking out, and I think that's not inappropriate. I don't think it's going to become the next pandemic. My guess is that if you could, sit down and have a couple of beers with some of these people, that they might agree the same thing, but they aren't going to say, "Oh, forget it, don't do anything; it'll all blow over." They're in a position of responsibility where they have to worry about protecting a population.
Yeah. Well, this is all about pandemic response and the whole system that's been built.
Right.
And it's gearing up, which is good.
It's easy for us. I can say it won't be a pandemic and be wrong, and nobody's going to come after me, right?
Well, we take the scientific view why we think it's not, and we've been talking about that. But these people are involved in public safety, public health.
Absolutely.
But we've even been pointing out we may be wrong.
Oh, of course.
That's happened.
Never.
Most of the time. But
I agree with Rich. If I were sitting in Nancy Cox's chair, I would be saying we need to take this seriously.
If I'm wrong, nobody dies.
Right.
That's true.
The thing is that I find, I don't know what the word is, ironic, is that, how many people in the U.S. actually get the seasonal flu vaccine? 40%, right?
Yeah.
So I don't know what the number is for a pandemic year. I'm sure it's not a huge amount more. In fact, the numbers are out there. I just don't remember them. But here we have, gearing up, pandemic preparedness, and then you go and you offer people the vaccine that you make.
They don't want it.
And most people don't.
That's right.
Well, but there was a very significant spike. I don't have the exact numbers in front of me, but I actually wrote up the meeting where the data were discussed, or a meeting where the data were discussed extensively. There was a major spike in vaccination for both pandemic and seasonal flu in the H7N9 year.
Yeah.
So people did rush out and get it. And the big problem that year, of course, was the supply, because everybody wanted the vaccine, and it took some time to get it out to people.
Let's see. I got the article here. Let's see if I can find the number. This is called Monovalent Vaccination Coverage U.S. 2009. And it's a lot of text here. Yeah. Oh, here we go. Percent vaccinated. January 2010. 20%.
20.
But that was as of January.
Yeah, so presumably went up
2010.
But of course, this thing started in April 2009. And of course, the vaccine wasn't available till August, September. Right. So I don't know what the final results were. That's an interim result.
So, Vince, who's the point person at WHO in charge of influenza surveillance?
You don't know.
Because I'd like to know what their opinion is about.
They have a great
they have a great site for it.
There's a great page at WHO, which we will link to.
That's my pick of the week.
You can't do that, dude. Let me go. It's called global alert and response.
Yes.
And then they have, human infection. They have updates every day pretty much. And then there's a link that says more on human infection. And on that page, they have a weekly report on the number of confirmed cases, which is what we talked about. They have those nice maps.
Yes.
So every week they put that out.
But do they think it's going to become a pandemic? Do they offer an opinion?
They say here, as there's limited information, but there's no indication that it can be transmitted between people.
Right.
WHO is very conservative.
Of course.
They haven't made any pandemic numbers. Have the different pandemic levels.
They got to be really careful. If they start using a word pandemic, people start flipping out, and that's
Yes.
and you have to be careful of that.
Yeah.
You're right.
I have a couple of observations, sort of miscellaneous observations. One is, I found it very interesting that in some of these really more science-based papers talking about these strains, there was frequent reference in discussing determinants of transmissibility, frequent reference to the Fouché paper.
Right.
Okay.
The H7N9 ferret experiment.
Right. Okay. So those ferret transmission experiments, even though are being accessed.
Yes.
All right. as part of the landscape that helps people understand the evolution of these viruses. So it's important that those experiments were done. It's important that they got published. The other thing that interests me is it seems to me that within the last decade or so, our ability to track and analyze these things has really increased incredibly. And I wonder if over the next 20 or 30 years, we aren't going to see an increasing number of these examples of some influenza invading the human population sporadically and then burning out. And our whole perspective on this sort of incident is going to be different.
I'll bet, yes. Same with the coronavirus, right?
Yeah.
This
right.
recent isolate in the Middle East. I think
And
that's
so-
part of that, We're looking more. Yeah.
So it's important to do this stuff, and maybe 20 or 30 years from now, with more information about genetic determinants for transmissibility and better epidemiological data that comes out of these sort of studies, we'll be able to, see one of these things happen and say, oh, that's another one of those,
Yeah.
Oh, it's another one of those. Interesting.
I think that, we'll have information. We'll get the sequences, and we'll go, nah, this is no big deal, because we'll know. But it's going to be a while, but that's the idea. As long as we can keep getting funded to do this research.
Ha.
I don't know what's going on with this SARS virus. I haven't heard anything for the last couple of weeks. Seems to have quieted down, as has the
Just the last one.
the Bunya virus causing severe fever with thrombocytopenia syndrome.
Right.
Transmitted by ticks. Now it was in Japan. We talked about that a while ago.
Well, there's in the regular news, there's really only room for one virus at a time. So that's why some of those stories have quieted down. But also in literature, we haven't heard a whole lot about those. So I think maybe there just haven't been that many more cases.
Yeah.
Don't you think, Alan, that flu really grabs the headlines more than any other virus?
It definitely grabs attention.
Why is that? Because it has this pandemic
Because everybody can
potential?
relate to it. Everybody has had some experience with it and has some thoughts about it, and
That's true.
and has at least a general notion of how bad it could be.
Yeah. And everyone has not had Ebola, of course.
No, right. There's that
Yeah.
gets headlines just because it's famous for being famous.
HIV gets headlines, but more in the lines of
The other thing,
vaccine
of course,
work.
Right.
this is an aging population. The United States has a very top-heavy...
You can speak for yourself.
No, I'm speaking for a whole bunch of other people, too. The AARP has a huge membership, and so that's the most susceptible group, mostly, to the mortality effects of influenza.
Did you know I'm actually getting younger?
I did know that, Vince. I can see that from here, actually.
I really
He no longer has to shave.
I like that movie. I wish I could figure it out.
Yeah, right.
Now, we had another story here, but maybe we should move on to email.
Yeah, I'm thinking
I
that's
think
appropriate.
I think we're over our time.
And then let's save for next time.
Yeah.
First, Rich, why don't you read the first email?
Okay, let's see here. I gotta scroll through this other stuff.
You could read the next one, Dickson.
My wife borrowed my iPad.
Right. Georg writes, "Hi, TWiV team. I discovered your netcast via Omega Tau, a science netcast by Marcus Volter Nora Ludwig, when you did a joint episode on viruses there, July 2011. Currently, I am at TWiV episode 191. I started from episode 1 in chronological ascending order. I also enjoy TWiV very much.
Oh, thank you.
I don't know if you are already aware of this netcast distribution site," and he gives a link to bitlove.org. Basically, it is a web service for making netcast distribution via P2P, BitTorrent, easy for publishers, while providing a comfortable directory for listeners. It is a community-driven project by netcast enthusiasts. It was started by a German developer who is connected to Tim Pritlove, thus the name, who is probably the most famous German netcaster. Omega Tau is already there, and he gives a link to that. I don't know if you pay much for your bandwidth or how much it is saturated when you release a new episode, but P2P technology would certainly help with that, i.e. saving your own bandwidth and improving scalability. Besides the technical advantages and interesting technical details, the bitlove.org directory has grown into a collection of great netcasts such that you probably would attract some new listeners there. I, for example, have discovered many nice netcasts there, which I would probably have missed otherwise. Best regards. And that's from Georg.
Wow.
Alan, don't you need to share on a peer-to-peer thing as well as download?
It depends on how it's configured.
Okay.
Yeah, it's usually people leave their P2P client up, and then it splits the file across a network of individual computers and sends them through. And this is used for transferring large files, large video files, high-resolution videos, that sort of thing. Yeah. I think you can use it without doing that, but you'd kind of be freeloading.
Yeah, that's the idea, right? You got to share what you download.
Yeah, you should pay it forward. So to speak.
Certainly there are people who we would probably gain as listeners if we did this. But our bandwidth is currently taken care of by the American Society for Microbiology, and for which we are very grateful. But I don't know if I could do both, but it would be an interesting directory to be in. So maybe we could make this available in addition to our current distribution network. Dickson, here's one for you. Laura writes, "Hiya, I am studying for a PhD in the UK, and I was wondering, are there parasites of parasites?"
Well, yes, we've had a discussion about that.
Yes.
Can you remind us of
double-stranded RNA viruses that infect Leishmania and Giardia.
And the other one?
And Trichomonas.
Trichomonas, yeah.
That's correct.
Giardia, Trichomonas, yeah.
And there are viruses of viruses.
Indeed. Mimi and Maxie.
And viruses of parasites.
Right.
So.
Right.
Yeah.
And so on ad infinitum.
Viruses of helper-virus relationships. Sure.
Sure.
What do you think, Rich?
Just about anything you can dream up exists.
That Jonathan Swift little ditty is really appropriate in this one.
I don't know any examples that you guys haven't already mentioned.
Yeah, there are plenty. And there are more that we don't know about.
There are. That's
I'm thinking of the Marilyn Rusick's plant parasite thing comes to mind, where you got viruses that infect fungi that infect plants, and the whole thing impacts on the health of the plant.
Of course, those are symbiotic relationships. It's not parasitic, right, Dickson?
No. Symbiosis is a general term.
I thought parasitic relationships were where one impacts the other negatively. That's what you told me.
I told you correctly, but if you think of symbiosis as a relationship term...
Yeah, but they don't hurt each other in a symbiosis, right?
Yes, they do. It depends on the kind of
They
symbiosis.
do?
So you would categorize parasitism as a form of symbiosis.
Correct.
Oh, okay.
Yeah, so you have mutualism, you
have
commensalism, and you have parasitism. And it's all part of the symbiosis.
All part of symbiosis. Symbiosis is just living together.
It's correct. They're two dissimilar organisms interacting.
So they don't have to be married.
Especially not today. You can have two similar organisms living together now.
Alan, can you take the next two?
Sure, yeah. The one from Randy is short. Love the podcasts. They've been entertaining and very helpful in my epidemiology class. Thanks.
Great.
No problem. We enjoy
Short
making them.
and sweet.
And then Catherine writes, "Hi, TWiV Star hosts. I'm writing from dark South Korea, where it's 46 Fahrenheit, 8 Celsius at 8 p.m.
We get good weather reports.
Yes, we do.
And
I don't have a question per se. I want to confirm a piece of knowledge that I hope I've picked up since TWiV 1. People, cells, cell cultures, and animals are all different. So take H5N1 flu. People react one way, anywhere from mild respiratory infection to death. In cells, you can have the H and the N working their magic, cutting off their hands, but just slowly enough, as Alan described a few TWiVs ago. Then there's cell culture. When you talked about lung cell culture and how it stratifies to resemble actual lung tissue, you might, please correct me if I'm wrong, but this is where I'm fuzziest, expect full-on virus production if using a human-adapted virus. And as we saw with the ferret experiments, animal models are not human. You may or may not see the same behavior. In any case, symptoms of the disease are likely to be different, weight loss in mice with flu. As an aside, you spoke of how things have changed in terms of techniques in your field. In my former field, infant perception and cognition, things haven't changed much since the 50s. Thank you for your time in reading this letter, and if it's off the deep end, feel free to send it to the electron recycling plant.
Electron recycling plant.
That's good.
No way it's off the deep end. This one, the key sentence here is that people, cells, cell culture, and animals are all different.
Different.
Absolutely.
And as we talked about at the top, even individual cell cultures of the same line are different.
Absolutely. Sure. And people need to keep that stuff. Scientists need to keep all that in mind.
Yeah, we actually discussed this a little bit at the top of the show, right?
We
This
did.
whole thing.
We did.
Speaking of weather, I visited Berkeley last weekend to do a TWiV, and my two hosts, Lisa and Zoe, met me at San Francisco Airport, and they had a sign saying, "Welcome, Dr. TWiV. It's 72 degrees and sunny." That was just great.
Yep.
Thanks, Zoe and Lisa and Emma and everybody out at Berkeley. That was a great trip. I really had a good time.
Yeah, it sounds like it.
Dixon, can you take the next one?
Nope.
Rich, can you take the next one?
I can't take it because I don't have an iPad.
I know. I love bugging you. I'm sorry.
I love being viralized.
Joseph writes, "Dear TWiV Mavens, as a chemistry graduate student at your university, Professor R., I took a cumulative exam that required us to fill in a blank periodic table, so it is not unheard of.
Nice.
Though Alan's point about lazy testing may well have played a role. The only hard parts were the metals for me. Any organic chemist should be able to fill in the main group.
That's true.
But to be a good inorganic chemist, it really helps to know your way around your section of the D and F blocks. The professor had given that test before, so we were able to cram for the day. I
Yes.
certainly couldn't do the D and F metals now.
Right.
By the way, my thesis advisor at Columbia, Ron Breslow, was a
yes.
Westinghouse Science Talent Search finalist in high school,
Ron
as was
Breslow.
I. All the best, Joe."
Wow.
Ron Breslow.
So
also
that sounds
got
like it was a real test of his mettle.
Yeah. Vroom. Joe is in securities these days. Sounds
Really?
like
Securities.
Sounds like you were really into science, though.
Yeah.
So, Alan, lazy testing means you don't want to make up an exam, so you just throw out that kind of test. Is that it?
Well, and you don't feel like grading an essay
Yeah.
or some other thing that would be difficult to grade, whereas you can just throw out blank periodic tables. Very easy to not only prepare, but very easy to check.
Right. Ron Breslow also, by the way, got the Presidential Medal for Science.
He was a fabulous chemist, right?
He's a wonderful
So let me tell you my Ron Breslow story. When I
Please.
first came to Columbia in 1982, I gave a talk to the Department of Biochemistry. And Ron Breslow came. I don't know why he came.
Oh, wow.
And I was talking about polio and how the protease cleaves the polyprotein, right? This is 1982.
Mm-hmm.
He said, "I'll bet you could make peptides to inhibit the protease and have antivirals." And I said, "Yeah, probably." So he
Bingo.
said, "Let's make some." And he made a few,
Yeah,
and I tested them,
yeah.
and they didn't work, and I just dropped it as a young idiot. But this was way before the protease inhibitors of HIV and Hep C today. I didn't get it. I was so young and naive. I didn't get that this great chemist was trying to reach out to me, and I should have pushed it.
Right.
We could have had the first protease inhibitors.
Yeah.
He was willing to make things and let us test them. I didn't get how cool that was. So you look back and, man, all this stuff you screwed up.
No regrets, Vince. No regrets.
No, I'm just venting my frustration here on TWiV. Rob writes, "Hi, my name is Robin, and you mentioned me on your podcast a few weeks ago in the context of the viral challenge studies that we conduct at Flu Camp. I wondered if you would like to visit and let me show you what we do and why it is so important. Thanks." This is in the UK, I think, right? Flu Camp.
Flu Camp.
I would. If I'm ever over there, Rob, I will definitely visit and
Don't
check it
sign
out.
up for Flu Camp, though.
Plague Camp. Stay away from Plague Camp.
Rob has at the bottom of his email, this email was sent from a mobile device with an annoyingly small keyboard. Apologies for any typos. That's cute. Should we do one more?
Sure.
Alan.
Judy writes, TWiV Gurus. First, thanks for all the time you spend on TWiV, TWIP, and TWIM. I've written before. I'm a high school science teacher in San Diego, California. Sunny, 19 degrees C. And wanted to write to you about two topics in this week's TWiV. First, if you're going to talk about science education, check out the Next Generation Science Standards, NGSS, the putative science standards for K through 12 grades under the Common Core Education banner. Gives an address, nextgenscience.org. Click on NGSS Standards button at the top of the page. The Common Core standards are standards that have been adopted in theory by 48 states, not Alaska or Texas, and are designed so that all the states in the U.S. will be teaching and testing the same material. So far, the English and math standards are being implemented, and the science standards are in their second major revision. If you look at these standards, you'll see that many teachers, myself included, are worried about the lack of basics required and the potential lack of rigor. For instance, stoichiometry will no longer be required for high school chemistry. It is recommended. Scary to think what will happen to kids in college bio and chem classes if they can't balance equations. I bring this up only because if you have a program in science education, please get people who are familiar with NGSS, as these are the standards that will guide K through 12 education for many years into the future, starting in 2015 or 2016, depending on when they're completed. Second, in episode 225, you also mentioned a need for basic cell bio podcasts and videos to help people understand some of the basics. Since I'm about to embark upon teaching honors biology and a biotech course in high school, I plan to start on a collection of just such types of podcasts or screencasts this summer. I've been trying to decide what subjects I should pursue, and since you have a collection of great minds and experienced professors, I wondered if you would give me some suggestions for topics you think would benefit both my students and the TWiV listeners. I'll be happy to share these with you if they live up to the standards of TWiV. I'm new at this, but I think it's an important way to reach students and individualize instruction. Thanks for all the ideas you've put in my head, Judy.
All right. I think we had talked about education, and probably we didn't know what we were talking about. So
Right.
she's telling us, if you want to do a show on it, here you go. So, yeah, I this is a great site. And I think harmonizing science is tough, right?
It's very difficult.
Yeah.
This has been tried several times before. There are efforts going back at least to the 60s to come up with national science curricula.
that's right.
The problem in this country is something called federalism, where each state is, as far as education goes, it's essentially its own kingdom.
Correct.
And each district within that state is its own fiefdom. So you're not just coordinating 50 states, you're coordinating a few thousand counties to get the curriculum out there, but this thing, I guess, has enough support that it may actually unify the curriculum to some extent. And then the question is, what do you teach? And how do you teach it? And how do you know that you taught it? And that gets into the whole testing issue.
The evaluation is very tough.
Virology.
Hey.
By the way, I wrote a piece on science education, did some reporting for the Journal of Clinical Investigation, of all places.
Is it open access?
It is, I believe.
Put the link in.
It's posted.
Yeah.
We'll put it in the show notes.
Posted.
Yeah, it's called It Takes a Village to Raise an Idiot.
Wow. Maybe you should be on our show on education.
It was several years ago. Let's see.
Judy should check it out.
Oh, other people have now used that title. No. Oh, there. Yeah, this was back in 2002. Not a whole lot has changed, probably.
So those are idiotypic, do you think?
Yes.
So I think you ought to make videos about basic cell structure, nuclei and cytoplasm and all the organelles, the microtubules and how things move around. You should cover the flow of genetic information from DNA to RNA to protein, how it gets out of the nucleus, splicing, transport of proteins up to the plasma membrane. What else? So basic cell biology stuff.
Yeah.
Control of gene expression at the level of transcription, DNA
But
replication.
you have a lot of intermediary metabolism to cover.
What's that? Do you need that?
I believe in order to make a cell work, you've got to have something
But
like
that's
that
not cell
going on.
biology, though, right?
Well,
The biology of the cell.
virology is cell biology gone
Judy,
wrong.
if you could do a bunch on those and make them really simple, that would be great.
Yeah.
And you can run them by us, and we can give you feedback and tell you what to fix and not to fix if you want. We'd be happy to help you. Okay.
This week
Well,
in
yeah, and
science
if you're
education.
looking for a structure for the podcast, you could actually go to one of the standard texts. Go to Lotus or Baltimore.
Yes.
And or I'm sorry, Alberts or Baltimore, and use those textbooks, molecular biology of the cell and molecular cell biology. Those are the college level texts and the standards in their field, and just obviously don't do the whole textbook, but use that outline and that structure that they have, because both of those books are well organized.
We had a listener recommend an Albert's excerpt a couple of weeks ago, right?
Yeah.
Yes.
So I would use that, right? Let's see if I can find it.
There's a lot of video on this also, by the way.
That was episode before last.
225, I think. No, it was in the email. Yeah, it wasn't a pick, so I'm not finding it quickly, but you can check it out. All right, let's do some picks. Dickson Despommier, do you have a pick of the week?
I don't. No, I don't.
Man, you are such a slacker.
True. This is all true.
You don't have a pick?
I'm sorry.
You knew that we have picks at the end of our show. You're sitting here for almost an hour and a half.
Do you have a shovel of the week?
Have you done any reading this week?
Lots of reading, in fact.
Well, is there something you want to share with us?
Most of the reading is the reading that I've written. I'm busy
You
writing
want us
something.
to pick your book as
No,
a pick of the week?
no, it's not out yet.
All
Don't
right.
do it. Don't
All
do
right,
it.
Dickson.
Don't do it. I'm sorry. I'm going to defer.
So disappointed.
I defer to someone else's pick.
Okay. Rich Condit, what do you have?
I have something that Grant McFadden sent me. It's from the scientist, and it's just a little video on the life cycle of dengue. And it's an animation. We've done a lot of animations, but I thought this was particularly good. Nicely scientifically accurate, and it tells the whole story of how dengue works.
It's nice.
Cool.
Howard Hughes Medical Institute.
Yeah, very good.
Cool.
Wonderful.
Yeah, I'm going to use this in my course. As long as it's right. I have to check it out.
I haven't looked at it in a while. But my recollection of it was scientifically it was very well done. It was right.
It's only 4 minutes.
Mm-hmm.
And, okay, I will not look at it now. Thank you. That's cool. Alan, what do you have?
I have. Let's see, what did I put up here? Oh, yes. So this is a podcast that was just started this week. It's the Nature Past Cast. Nature has a few podcasts that they do, but they just launched this one, and it is going to be a science history podcast.
That's nice.
First episode is online, and it's about April 1953, which is Watson and Crick working on the double helix. And it's filling in some context from that time and some stuff that people may not know about that history. I think it's about 12 minutes long. It's not a huge time commitment, and I don't know how often they're going to be releasing these, but it'll be periodic episodes of it in the future, I think.
It's a good idea.
Yeah.
Yeah.
I don't like the title, though.
Well,
They could do better than that, Nature. Come on. Our friend Trudy had started a History of Science podcast, and sadly it has lapsed, but Trudy, you should go back to it because you're a scientist.
Yep.
All right. I have a pick, which is an art site, and I discovered this artist in Berkeley. Her name is Deborah Skloot, except it's spelled Sklut. I asked her if she was related to Rebecca Skloot, and she said maybe. I said, well, why don't you make a shirt with a HeLa cell on it and send it to her? Anyway, she makes shirts with phages. My daughter wore one to school this week. And Geiger counters and hearts and bacteria, copulating rabbits. Sorry. Yes, in fact, there are 2 rabbits. It's called Bunny Loving. And it's not bad, Dickson. Come on.
I didn't say anything.
You sound so critical.
No.
A gloved hand wielding a pipette, a microscope, an Eppendorf centrifuge, a brain, and rabbits again. She likes bunnies. Anyway, support her. Check her out. She was there at the meeting selling T-shirts. And actually, I did an interview with her, a video interview, which I will put up soon, where she explains what she does and how. And she has a couple of cool prints. She has one of T4 phages attacking E. coli, which is gorgeous.
Yeah, I really like that.
And that was in the office of Britt Glounsinger, who was
It's good. It's
a scientist that I had on TWiV last week.
I love it.
Her husband is a virologist, so she gets inspiration from him. So
I
check
think
it
I
out.
probably ought to get the T4 Phage T-shirt.
Yeah, my 2 kids have them, and they wore them this week.
Nice.
They're pretty cool.
It's a very cool shirt.
It is.
You've embarrassed me into.
I'm sorry, I'll cut it out. I won't publish it.
No. Please keep it. But I do have a pick.
It's only because I really like you.
I know that you don't pick on people you hate. You
I
don't
don't
waste
bother.
your time.
Yeah.
So my pick of the week is called a website, and it's called Agritecture.
Okay.
Agritecture.
See, there you go, Dickson.
I
It's
know.
easy.
It's very easy. I mean, I didn't have to relate necessarily to viruses, but this is run by a student.
Oh, that's your student, yeah.
Yeah, I'm mentoring a student at the Earth Institute, and all by himself, he came up with this this
mentoring a
wonderful. I'm mentoring a student, and I've
I
mentored
feel
men.
badly for him.
He's doing great, by the way. He's doing terrific work. But it's his name is Henry Gordon Smith, and he's got this fantastic accumulation of iterations of what vertical farms may look like, what they do look like, and what they should look like. Mm-hmm. And it's called Agritecture. I love the way he's put these two words together to form a visual image of the combination of farming and buildings. So I recommend it. He's a very clever
There it is.
person. Very, very clever
Buildings
Cool.
that grow food.
Cool, there you go.
Not bad.
I'll like
Not
it.
bad.
So I take it all back. I'll do more reading. I'll try to be more.
It's not hard to come
Creative.
up with a pick, Dixon. This is perfect, okay?
No, it is.
Just share something you like. That's the
Well,
whole
you asked
point.
me if I was busy. I was, and that's the kind
Okay.
of stuff that I'm busy with.
We have a listener pick of the week from Mike, who writes, "Hello, folks, from an avid listener. Towards the ends of episode 224, there was a discussion of the periodic table of the elements. For a while, I've had chemistry in its element from the UK's Royal Society of Chemistry in my podcast subscriptions. When the series began, the presenters went through the periodic table, giving a 5-minute or so summary of the element's history and uses. Since reaching element 118 in 2010, the podcast shifted to chemical compounds, keeping the weekly release cycle.
Nice.
If you only want the original series, here's the link. There's an iTunes feed, etc. And then he gives links for the series on chemical compounds. If you read this email on a show, just call me Mike in Florida rather than trying to pronounce my last name. 57 Fahrenheit, 14 Celsius, now in Daytona Beach with clear blue skies. No, you don't need the weather. You just need TWiV.
Love
That's
it.
right.
Just love it. You know,
Except that weather is probably a month old.
Probably the same, though, right?
That's okay.
History
no,
of weather.
57 Fahrenheit? No, it's warmer than that in Daytona Beach
Daytona
today.
is where?
It's on the coast.
South Florida on the coast.
South Florida.
which
yeah.
coast?
South Coast.
The Atlantic
East Coast.
coast. About
The East
the Atlantic coast about it's actually midway north and south.
So, Dixon, when you're on the very south tip of Florida, when does
Yes.
it become east and when does
That's
it
a
become west?
very good question.
Must be from the middle on either side.
When you turn north of or when you turn east of 180 degrees, I guess.
It's a
Yeah.
mathematical point.
I've looked at both of these links that he has here for both the periodic table and the compounds, and they're really very nice. It's very
Yeah.
well done.
Hey, why don't we go through a periodic summary of the viruses?
Hey,
In a couple of episodes.
at least alphabetize them.
Could do that.
There'd be a lot of
There was
viruses.
There was a classification 101 a long time ago,
There
right?
was.
Yeah,
There
that's true.
was.
No, we should get back to 101.
We
What's
should
next?
do that.
We just did splicing, right?
Yeah.
Right.
We were going to do one a month or something like that.
Yeah, right.
What happened to that?
Yeah, exactly.
Well, Vincent started moving the schedule of TWiV around and going on his world tour.
Sorry.
And
exactly.
Yeah.
It's all his fault.
he's too famous.
I'm sorry. We will do it soon. Okay, I'll get on the schedule and do it. Anyway, thank you, Mike. That's cool.
Hey, Vincent.
Yeah,
Vincent.
Yeah.
It's okay.
Yes.
It's okay.
I'm surprised he didn't pick Tom Lear's version of the
Sure he
periodic
knows
table.
about it. I've always wanted a chemistry podcast, so I'm going to subscribe to this. I love chemistry.
Yeah, this is good.
Totally agree.
Thank you, Mike. You can find TWiV on iTunes and at twiv.tv. And if you like what we do, go over to iTunes and give us a rating and subscribe. That would be really good. It helps to keep us prominent in the directory over there, and that way many people will learn about viruses, which is what we want to do. Send your questions and comments to TWiV at twiv.tv. Dickson Despommier is at verticalfarm.com, Racaniello.org, and medicalecology.com. Thank you, Dickson.
Welcome, Vincent.
I didn't mean to shame you.
You didn't. It's impossible.
Well, you got a pick out of him. That was good.
He did.
I like the pick.
He picked on me, so I picked back.
I just think you have plenty of things to share, that's all. But thank you for joining us, and I appreciate you coming in so early.
I love to do this. I do. So
Okay. Thank you very much.
glad to be a participant.
Rich Condit is at the University of Florida, Gainesville. Thank you, Rich.
You're quite welcome. Always a good time.
And Alan Dove is at alandove.com and on Twitter. Thank you, Alan.
Always a pleasure.
I'm Vincent Racaniello, and you can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.